right, so this audio lecture is going to talk about antigen recognition by T cell lymphocytes. So the first portion is going to be about T cell receptors. How are they different than B cell receptors? How are they the same as B cell receptors? And then the second portion is going to be about antigen processing and presenting. So when we look at the properties of the T cell receptor or TCR, it is very similar to the Fab fragment that we see in the B cell receptor. Okay, unlike the B cell receptor, it doesn't get secreted, so it's always membrane bound, and it only has two chains. So remember that the B cell receptor is made up of 4 chains, 2 heavy chains and 2 light chains. For the T cell receptor, there's just 2 chains. They are heterodimer, so there's 4 genes, loci that make up a T cell receptor, and the T cell receptor is going to select 2 of those genes to be expressed. So either alpha beta will pair, or you'll get gamma delta pairing. It, like the B cell receptor, has a constant region that's pretty conserved, and then we also have a variable region, and it's in the variable region where we have our antigen binding site. Unlike the B cell receptor, which has 2 antigen binding sites per receptor, the T cell receptor only has a single antigen binding site. And the T cell receptor is going to be able to interact with MHC class 1 and class 2. And we'll look more at that when we look at presentation. So you can see here in this diagram on the left, the image of a T cell receptor compared to an antibody, or also known as a B cell receptor. And so again, some of those comparisons we were just talking about, it's going to be membrane bound. So there's going to be a transmembrane region that you can see there. And again, it's going to have 2 chains. The classic dominant pairing that you see is alpha and beta, because that's kind of the default that the T cell starts with. You'll notice that it also has sites for carbohydrates to bind to for modifications. And again, you can also see that there is the variable region where you have your antigen binding site, and then your constant region also present. On the right-hand side, we have a ribbon model of the T cell receptor, and you'll see that it has these what's called the CDR loops that make up the antigen binding site. So you can see on the top of that where the variable region is, it has these loops, and again, those are called CDR loops. To have a functional T cell receptor, you have co-receptors. So CD3 plays a critical role in T cell activation because it's required in order to kind of think of stabilizing their interactions for the T cell receptor so it can interact with the MHC. There's additional molecules that are also involved in those interactions. And later on in the semester, when we talk about activation, again, those will become critical. You can also see that there's signal transduction molecules that are going to help with cell signaling events that would occur if the T cell receptor binds antigen that's being presented to it through an MHC molecule. As I mentioned, there's 2 different pairings or classes of T cell receptors. So you can see on the left-hand side, we have alpha and beta, and then on the right-hand side, we have gamma and delta. Similar to the B cell receptor, they have gene segments and they undergo rearrangement. So gene segment recombination also occurs in T cells, similar to in B cells. For T cells, we're going to have the alpha chain is going to get undergo rearrangement, and also the beta chain would undergo rearrangement. And so you can see how when there's the rearrangement that occurs, you're going to have the V and J joining for your alpha chain, and then for your beta chain, that actually has a diversity region. So that's going to have 2 rounds of recombination where you have the DJ being selected and then VD being selected. That ends up making up your final T cell receptor protein. And again, just like with a B cell, that means that T cells in the germline, their germline DNA is different than their DNA after they go through development and select their T cell receptor. So this is showing you the gene segments for the alpha delta, and gamma. It would be nice if they put in beta also in this figure, but they didn't. But for delta, alpha, and beta, they have variable diversity in joining and constant segments. For gamma, they do not have the diversity segments. So similar to the heavy chain or similar to the light chain of the B cell receptor that lacks the D segments. And then you'll also notice that there's 2 C gene segments, and each of those have their own J segments. So there's not quite the same that we see for the light chain where there's the pairing, but if you end up with certain joining segments, you're going to have certain constant segments. So if we look at the differences in diversity between the B cell receptor and the T cell receptor, we can see that we have the different segments, right? So you can see the number of segments for variable segments, D segments, and J segments for each of the different chains. Then if you look at the V gene pairing, the junction diversity, and then the final total diversity, because again, keep in mind that you have heavy chains and light chains pairing for the B cell receptor, and then for the T cell receptor, they're looking at alpha and beta pairing. You can see there's actually more diversity in the T cell receptor, and that's really important because later on, when we're talking about development of T cells and B cells, we're going to talk about a process of negative and positive selection. And so with T cells, they undergo both negative and positive selection. For B cells, they only undergo negative selection. Positive selection is where you make sure the receptor actually binds MHCs. And so T cells have to go through this additional selection. So a lot of the diversity might end up being lost because you might make combinations that can't bind to MHCs. And again, T cells require presentation through MHCs in order to see their antigen. So having a higher amount of diversity to begin with allows therefore there to be a deletion of a good number of them, and you still have a good repertoire of T cells that will protect you. Next, we're going to look at antigen processing and presentation. So again, as I've mentioned several times already, antigens need to be processed and presented on MHCs, MHC class 1 or class 2. So T cells cannot become activated on their own. They need to be presented antigen. So again, keep in mind the B cell can be out in circulation and it doesn't have to have processed antigen presented to it. Its B cell receptor can interact with that pathogen, that cell intact, and recognize its epitope if it's an extracellular epitope, okay? But T cells can't do that. So T cells aren't directly interacting with the antigen, it's what's being presented to them, either in class 1 or class 2. Now, the reason why there's these two classes is class 1, MHC class 1, is going to present internal antigens, and that's if like the cell's virally infected, it's going to allow it to process those antigens, those proteins in them, and then present them and kind of do a check, like, is this okay or is this not okay? Okay, there is a T cell that sees this. Okay, this is not okay. The way the processing happens for MHC class 1 for internal antigens is through the proteasome. Okay, so we'll talk a little bit more about that. For MHC class 2, this is antigen that's been processed through phagocytosis. So you'll remember with phagocytosis, anything that's engulfed ends up getting digested when the phagosome fuses with the lysosome. Digestive enzymes breaks it up into little pieces. Those little pieces can contain potential epitopes, right? So some of that material gets put onto MHC class 2s and then gets presented on the surface of that phagocyte. So phagocytes are going to have MHC class 2 so they can present to the T cells. So this is an oversimplification of what's going on here for processing and presenting, but the idea is that we've got some kind of protein in the cell. It's going to get broken down into it's going to get broken down, and then it gets presented an MHC molecule, and then gets presented to a T cell. Huge oversimplification. So I would say this level of understanding might get you a C response at best. There's a lot of additional details that are missing from this, and you're going to see other figures that better demonstrate with detail. We don't even know whether this is MHC class 1 or class 2, what kind of T cells being presented to, how did it get digested, what kind of protein is this, was this something that was because of viral infection, or was this something due to phagocytosis? So again, it's just meant to be a kind of an overview of like, oh, this is what's going to happen. But so if this is all you study, please don't. So as I've mentioned, there's two different MHC molecules, class 1 and class 2. So you can see the difference between the two. So one of the key features, if I was to ask you to identify them, is to look at the transmembrane domains. You'll notice class 1, like a Roman numeral 1, only has one, where class 2 has two transmembrane domains. Okay. If you also look at this image doesn't really nicely show it. It's actually our next figure that will show it better. But if you also look at the domains that make up it, you can also distinguish the difference. So we'll look at the next figure. So in this figure, again, you can see MHC class 1 on the left and MHC class 2 on the right. So you can see in class 2, there's two chains. There's an alpha and beta chain, and each of those have two domains. So you'll notice there's alpha 1, alpha 2. So two domains, beta 1, beta 2, two domains. And again, they're going to have two transmembrane domains that attach it to the cell. MHC class 1, you'll notice that there's three alpha domains, alpha 1, alpha 2, and alpha 3, and then there's one beta domain. And so their four domains are made up different, so it's not an even sharing of domains between that. And the beta domain doesn't have a transmembrane portion, okay? The binding grooves are also different between class 1 and class 2. So the area where the peptide or epitope antigen binds to is known as a peptide binding groove. And you can see on class 1 how that peptide actually has to fit in there. So class 1 is a little bit more like a cup that has closed ends. So it needs to be able to fit into that groove and it can't hang over. Class 2, the ends are a little more open, and so there's a little bit more freedom, if you will, in what fits in there. There's anchoring points, some of the side chains anchor and hold certain amino acids in position, and those become critical in the recognition, but sometimes there's a little bit more wiggle, if you will. So certain substitutions in amino acids aren't as dramatic in whether the peptide will bind or not. And peptides binding in class 2 tend to be a little bit longer again because it's not that closed end. When we look at the distribution of MHC classes, we will find that virtually all cells have class 1 except for erythrocytes. So we mentioned this in unit 1. So red blood cells do not have class 1. So erythrocytes, red blood cells, are responsible for producing hemoglobin so we can transport oxygen. That's what they do. And so they're not playing a role in making sure everything's okay because they're focused on their job. Other immune and non-immune cells are going to express class 1. So you can see that, virtually any other cell in your body is going to have class 1. Now, immune cells have a higher expression of class 1 than compared to your normal cells because they're going out and they're trying to find, that's their function, that's their job. So they want to make sure they're expressing class 1 so that they can interact with other immune cells and say, hey, this is what's going on. Came across this virus. Let's get rid of it. When we look at class 2 is produced in immune cells. So you'll notice that cells that undergo phagocytosis, like macrophages and dendritic cells, have high expression of MHC class 2. Now, in your table, it notes that neutrophils don't have class 2 expression. That's, and this is where I'm going to disagree a bit with the textbook. So neutrophils can express MHC class 2 under certain conditions like inflammation. So a lot of times what happens is the neutrophil is going to engulf and then they get engulfed by a macrophage. So the macrophage is the one that ends up doing the presentation. So it's kind of a piggybacking that happens in that presentation. So that's one way that if they're not expressing it initially, that antigen is still being able to be presented. So in addition to CD3, there are additional glycoproteins that are important that act as co-receptors, and so there is CD8 and CD4. So you guys know about those as markers for T cells that we can differentiate between different T cells. We saw that in our first journal club paper. So CD8 is 2 chains, alpha and beta chain, and then CD4 is a single chain, and so they're actually going to interact with the MHC to stabilize the interaction. And so we'll look at that in the next slide. So in this slide, you can see in that first panel how a CD8 positive T cell, it's interacting with a virally infected cell with an MHC class 1, and you can see that CD8 is stabilizing that interaction so the T cell receptor can interact with the MHC and see, hey, is that peptide that's bound in its peptide binding groove the same antigen that I recognize through my T cell receptor? If it is, then that CD8 T cell, because it's a cytotoxic T cell, is able to actually kill that virally infected cell. Now, in our other panels, we see CD4 T cells interacting with MHC class 2s. And again, CD4 is stabilizing those interactions. You can see how that CD4, that single chain, is interacting with the MHC class 2. And so again, just like with our CD8 T cell, our CD4 T cell is determining whether it is able to recognize through its antigen binding site on its T cell receptor, whether it's able to recognize the antigen that the peptide that is in that peptide binding groove of the MHC class 2. Okay, what can happen in these cases is that CD4 T cell, which is a helper cell, it can produce cytokines, and those cytokines can then lead to activation of the immune cell that is holding the peptide. So in that middle panel, the T cell is interacting with a macrophage, so it's producing cytokines, then that leads to the macrophage becoming activated, so it's going to be able to kill more effectively bacteria that it's out there, that it's engulfed, or will potentially engulf in the future. On the right panel, we have a CD4 T cell that's interacting with a B cell, and so if it is able to recognize that peptide that's being shown to it on the MHC class 2, then it will start to produce cytokines. Those cytokines could tell the B cell to differentiate and become a plasma cell so it can secrete antibodies. So again, the interactions that are happening between these co-receptors and the T cell receptor and the MHC class 3, you can see in this figure. Again, the MHC, depending if it's MHC class 1, this is the target cell, so the CD8 cell, the CD8 T cell potentially is going to kill that cell. Okay, so that antigen, that epitope that's being presented in that binding groove, again, came from an internal source from that target cell. For the CD4 T cell, it's an antigen presenting cell that's presenting the antigen. And again, these would be phagocytes. This could be a B cell. This could be a macrophage, a dendritic cell that is taking material that it has taken up from a pathogen, from a dead cell, and it is showing it to the T cell through the MHC class 2, and that T cell is interacting through its T cell receptor. And again, these interactions are being stabilized by those co-receptors. All right, so this is like the big slide. This is this counteracts that oversimplification slide. So this is truly what's going on, in the processing of antigens, and whether it's class 1 or class 2. So we'll talk about class 2 first, because you guys are familiar with phagocytosis. So again, class 2. So one thing to keep in mind, this is not necessarily on the same cell that this process is happening. Obviously, on immune cells that express both MHC class 1 and class 2, this process can happen for both of these pathways. So for MHC class 2, again, we're talking about phagocytes. So what we would have is some kind of external antigen being engulfed, internalized, and then we're going to get the production of that phagolysosome where there's fusing. And so it's after the digestion that then now we have these MHCs that have been produced. So you can see, if you kind of backtrack through the ER, you can see the production of the MHC class 2 going through the Golgi, going out into a vesicle. That vesicle is going to fuse with our phagolysosome so that MHC class 2 are present there. That can get loaded in. We're going to talk in a little bit more detail about how do we stop peptides from being loaded in prematurely before it is introduced into the phagolysosome in just a moment. But that once it's loaded, the MHC class 2 is going to move to the surface where it can present and again interact with a T cell and potentially activate that T cell. T cell can produce cytokines, proliferate, and then go on, if it's a helper cell, go on and help other cells. For MHC class 1 on the right-hand side, you can see that there's this intracellular antigen. So again, maybe this is a virally infected cell. Maybe this is a damaged cell. And so it's producing components that signal that it's damaged. Those antigens are going to go through the proteasome. Proteasome is a cellular or protein complex that breaks down material into fragments. One of the purposes of that is to be able to reuse those fragments. Another purpose of that is so that it can then be loaded in. So those fragments actually enter into the ER, and we'll talk more about that process. But the peptides that are created through the proteasome are going to enter into the ER, and then they can get loaded in MHC class 1. So MHC class 1 synthesized, it's present in the ER, peptides get loaded, it moves to the Golgi, and then goes into a vesicle, so it can undergo exocytosis. So it's going to move to the surface where it can present. So MHC class 1 can present that peptide that was loaded in the ER to the surface. So some of the important distinctions here is what is breaking down the antigen, where is the MHC being loaded with peptide, and then the other piece is how do we stop the peptide from incorrect peptides from being loaded into the MHCs. So those are kind of three pieces that you should be able to identify the difference between MHC class 2 and class 1. So we're going to talk first a little bit about more about class 1. So here in MHC class 1, again, we have our protein. It's going to go through the proteasome. You can see on the right-hand side what the proteasome actually looks like. It actually has two caps and a chamber. So the protein's going into that chamber, and it is being digested down and into fragments. Those peptide fragments go through a structure called TAP. So it is a kind of transporter that can open and close, and it allows for the peptides to enter into the ER. So once they enter into the ER, they can interact with MHC class 1. Now, the proteasome is kind of, randomly cutting, and so the fragments that are created can be different lengths. And so when the peptide gets loaded into MHC class 1, if it's too long and if it's falling out, because remember MHC class 1 has closed ends, there's a protein ERP, E-R-A-P, that will actually remove it's a protease that will remove amino acid residues, so it will then fit into that MHC class 1, okay? Because remember, MHC class 1, the peptide needs to fit into that groove. It can't hang over the groove. So there's a number of proteins involved in the process of loading the peptide into the MHC class 1 groove. So just to get MHC class 1 formed, calnexin is going to be critical in that stabilizing the MHC class 1 so that the beta subunit, the beta domain, can bind to it. Then calreticulin and ERP57 are going to block the MHC1 molecule so that peptides don't accidentally load in, or a protein doesn't accidentally load in. So once that protein enters through TAP, so you can see how they're entering through TAP through that, so degraded by the proteasome, enter through TAP, and then they're allowed to go and load in. We would then have any kind of editing, removal of amino acids that need to be done. And then once that's been completed and we have the releasing of the MHC class 1 by calreticulin ERP57, that's going to signal for the MHC class 1 to go into a vesicle so it can be exported out of the ER and then to the cell surface where it can be presented. Now looking at MHC class 2, again, keep in mind it is going to be synthesized, made in the ER, but it needs to get into a vesicle and get to the phagolysosome without having peptide or proteins that shouldn't be loaded into its groove present. So the invariant chain is a protein that actually blocks that binding groove so that peptides that might be present in the ER don't accidentally load into them. In its journey to the phagolysosome, when it's in the vesicle, that invariant chain gets cleaved, and what's left behind is just a little fragment called clip. So once it fuses with the phagolysosome, clip will dissociate, and then that allows for a peptide to then bind to MHC class 2. DM is the protein that facilitates the release of clip that then allows a peptide to bind, and so then when a peptide is bound, the MHC class 2 can move to the surface where it can undergo presentation and interact with a T cell receptor. So unlike the B cell receptor and the T cell receptor, MHCs are co-dominantly expressed. That means that you express both MHCs from maternal alleles and maternal alleles. So both from your mom and dad, your immune cells are going to have MHC class 1s from both of them on all of those cells that have MHC class 1 and class 2. So B cells and T cells, they're only expressing one of those alleles to make either your paternal alleles or your maternal alleles to produce your receptor. This allows for more diversity of the MHCs that are on the surface of the cell, because remember, with each of these MHCs, they have the peptide binding groove, and so each of them are going to have little variations. So the different isotypes, allotypes that end up being produced allow for additional different peptides to be presented. And you want MHCs to be able to present as many peptides as possible to then allow for the T cell to determine, okay, do I have the T cell receptor that recognizes that peptide? So it's the limiting factor of having a response is really the T cell, not the presenting cell, not the antigen presenting cell, right? Because they have a wide range of HLAs. There can also be polymorphisms. So polymorphisms where we have these like single nucleotide changes, so they can lead to a single amino acid change, which leads to a side chain change. So that can affect what peptides can bind in the groove. And so here you can see how certain sites are important. And so where we see those red dots are critical to binding of the peptide in the peptide binding groove. So when those change, they change what can be bound. Because again, when you change amino acids, you change the side chains. And when you change the side chains, you change what can bind. And if you change what can bind into the peptide binding groove, you're changing what peptides can be presented to the T cell receptor, right? And so polymorphisms are really actually a good thing because it increases the diversity of the peptides that can be presented. So this figure shows you what's been shown in populations. So when we have epidemics of infectious diseases, there's usually an expansion of the number of MHCs present because of polymorphisms. They actually end up getting kind of selected for because there's a more diverse number of organisms that you're being exposed to. And so you are presenting these different peptides that are binding to those peptide binding groups. After post-pandemic or infectious diseases, what we see is that then there becomes a selection. So you're not then being exposed to these all these infectious diseases. And so when there's not the need to present those peptides, some of those polymorphisms kind of deselect out. Then if we have another emergence of infectious disease, you can see how there's this expansion back up of MHC polymorphisms that are being selected for. And then when after, then you see a decrease in the number of the different polymorphisms present in the population in the MHC. So again, you kind of see this expansion and reduction and expansion reduction. So it'd be interesting to look at, like with COVID and the pandemic, how that affects the polymorphisms within the population. I'm sure there's researchers who are looking at that. There's definitely researchers who have looked at the B cell response, the T cell response, and responses by innate cells and antigen presenting cells. So I'm sure there's papers out there or they're in works of progress. So if you do have any questions on any of the concepts that were presented in this audio lecture, please let me know. I'm more than happy to expand upon them during class time. All right, have a good day.